In the last lecture, we started talking about clustering and K-means clustering. So we're going to talk more about it today. So as a recap of the K-means clustering, it is an iterative algorithm, a very useful and successful algorithm that's still widely used today. So there are two steps in the iteration. The first step is to update cluster centers given the current assignment. Basically, we had a hard assignment. Every data point is assigned to one of the K clusters. Given the assignment in the first step of iteration, we will update each cluster center to be the average of all the points in that cluster. So that's where the name K-means come from. And mathematically, we can justify this update by showing that the mean is the minimizer of the sum of square distances when we define the distance to be the Euclidean distance or the L2 norm. And then the second step, given the cluster centers, we would update the cluster assignments by reassigning every point to its closest center. So it's possible that some cluster will stay in its original cluster, but other points may be moved to another cluster. So we do this iteration until the cluster assignments no longer change. So we said that this algorithm can always guarantee to converge to a local minimum of its objective function, which is this one. It's a function of the n assignments and the k centers. However, there's no guarantee that this function only has one local minimum. In that case, we had to try multiple random initialization to make sure that we had at least some of our results converge to the global minimum, hopefully. That's the K-means. And as I said in the homework, I want you to show why each step in the iteration can decrease the objective function, given the assignment fixed, why updating the center can decrease the objective function value. Then why given the centers, updating the assignments can decrease the objective function value. So later we talk about the K-medians. And the K-medians has a different distance. Here the distance is defined as the median. Okay, it's defined as the L1 distance. That is, we define the distance to be the sum of the absolute differences in each of the p dimensions. So it's not like the Euclidean distance where with each dimension we take the square and then we take the square root out of the sum across dimensions. Here we don't have that. We just have the absolute value in each dimension. And the minimizer of this L1 distance is known to be the median in each dimension. So this is called K-medians, which means we define the centers based on the medians instead of means. And there's another variant of the K-means algorithm called K-medoids. So this variant by name, it's different from the K-means or K-medians in that it uses another different way to define the centers. So here, the center of each cluster is, okay, let me change the pane. In each cluster is defined as a data point in that cluster. This is not commonly used because usually we don't care what the center is. But in some special applications, if you really want the center to be one of your data points, not an average, not a median, then you can use this algorithm. So it's still an iterative algorithm. You still have two steps. The only difference is that in the center updating step, let's say in the cluster, you have 10 points. You want to pick one of the 10 points as the center so that its distance to other points is minimized. So that's just the change. So in other words, you can think of it as adding a constraint to this updating step here. Here I'm searching for any p-dimensional vector to be my center MK. So in that K-medoids algorithm, your MK must be one of the xi's whose Ci is equal to K. So you are only searching among those points and to pick one of them as the center to minimize this objective. So it's a constraint objective optimization problem. Okay, so that's all I want to say about the K-means and these variants, K-medians and K-medoids. So today I'm going to talk about a key question in K-means algorithm. That is, how do we choose K? This is often a very practical problem in data analysis, and it's usually a general question for any unsupervised learning algorithm. For clustering, we need to decide the number of clusters. For dimension reduction, we need to decide number of dimensions we want to reduce our data to. So these are all pre-specified parameters as an integer. So here for this K, it is needed for K-means algorithm to run. Then how do we choose it? First, let's think about some intuition. So if you set a large K, then compared to a small K, the large K will give you more clusters and each cluster will be tighter, right? So that's the intuition we'll have here. So we know that if we go extreme, we can divide our data points into n clusters, 1 point per cluster. Then every cluster becomes perfect, right? There's no variance within cluster, just 1 point. And on the other extreme, we set K equal to 1. That means we don't do clustering at all. We just have one cluster. So therefore, there must be some trade-off in the middle. And we just need an objective function for us to find this trade-off. Okay, so one common criterion is called within cluster variance. Okay, so this is defined as WK, we call it. So there are two ways to write it. So here, let's just use one way to write it first. So we take the sum for cluster from 1 to capital K. So given a fixed K, we can calculate WK as the sum across clusters. And within the lower K cluster, we will calculate the average square distances as 1 over 2 NK summation. Okay, so here I'm just going to change the notation a little bit different from what we used before in the previous lecture. I will call the set CK as the set of points in cluster K. Okay, so this is defined as the set of data points in cluster K. And if I use our previous notation, it's actually equal to the set of the indices I such that the lowercase ci equals k. So we use that as an assignment, right? I from 1 to n as the assignment. And here we are just using the uppercase C for sets. So this is the set of cluster K. And then we know if we take the union for all the Ks, CKs, this will give us all the data points. So the union will be just 1 to n. Okay, so given this set for data points in cluster K, I can take the any ij that are in cluster CK. And what I can do is I calculate their Euclidean distance and square it. Okay, so this is how we define within cluster variance. And also here we can say the size of CK equal to NK. So that's the number of data points we have in this cluster K. So this is a way to actually define the within cluster variance. But one thing I want to say is that if you think about the 2, where does it come from? So here the 2 is because here we are not specifying which is I, which is J. So we could swap them. So I becomes J, J becomes I. So each pair of 2 data points will be counted for twice. So this is one way to define it. Or another way to define it, you can define it just based on the cluster centers. And I'm going to write it down here. So this formula and the other one is not exactly equal to each other, but both can be used. Is that we can define this as the average NK, and then we can define that for I in CK, I compute the distance between each xi to the center. I can also define it this way. So if both are actually used in books, and we can use this one to mean, okay, on average, to mean on average, how large are or how variant each cluster is, or how noisy data points are within each cluster. So this is measuring that. But as we said, our intuition tells us that WK goes down as K goes up. The more clusters we have, the smaller WK we would have. Because you can imagine, if I set K, capital K equals to N, then each NK is 1, and I will have 0 here. So WK will go down to 0. So this motivates us to choose to use the first method for choosing K. This is the graphical method, which called elbow method. It's very easy to use, but it's very intuitive. So that's why many people use it for justifying their K. So what it means is that we simply make a plot with K in the x-axis. Of course, it's an integer. So we have 1, 2, 3, 4, 5, and so on. And then we plot the corresponding WK in the y-axis, and we will track the trend. So for example, if we see there is a sharp drop from 2 to 3, and after 3, the decrease is not very obvious, then we will call this as an elbow, and then we will pick 3 as the number of clusters. So this is a very simple way for showing that. But usually it's not that obvious, right? So therefore, people may try to plot log WK. You know, when we take the log transformation, what we do is that we make those extremely large values not that extreme. So we can focus more on those smaller values. So sometimes when you do this, you can see the pattern more clearly. Okay. Or sometimes you may just take the adjacent to log WK. So you may do log WK minus 1 minus log WK. And we know this number must be positive because WK is decreasing. So if you see that you have a maximum at 3 and then goes down, then this gives you a clear picture to pick 3 as your K. So this is the easiest one and still the most popular one, I would say, in practice for choosing K. And the second one is also a graphical tool, and it's also widely used. It has an R function for implementing it. It's called silhouette. It's from a 1987 paper who proposed this graphical approach. So what this does is that it defines a metric to compare between cluster distance within cluster distance. So the ideal cluster, an ideal case should give us small within cluster distance, but big between cluster distance. That's the intuition. Okay, so let's just tell you the definition for using notation. So let's say, suppose we have X or I should say I in cluster CK. Okay, I'm going to use a notation to be consistent with myself here, but it's a little different from the notes, just for your information. So I need to define the following terms. The first one I need to define is what I call AI. So the AI is defined as the average distance of I to other points in CK. So in other words, mathematically, I can just write this as, given my previous notation, I can write it as nk minus 1, right? I have nk minus 1 points in cluster K other than I. Summation j in cluster CK, j not equal to I. Then I would have the norm distance between xi and xj. So this is how I can define my AI mathematically. And I also need to define distance between my point I and other points in, not in cluster K, in other clusters. Okay, so let's say that I would have this D, I write D, I call it K prime. Okay, so K prime is another cluster. And I can define this to be, or I should say, maybe CK prime, that's better. Okay, CK prime is another cluster. So I will define this as the average distance of I to cluster CK prime. And how do I define it? If the average is going to be taken for all the points in cluster CK prime. I should say it to all points in cluster CK prime. So mathematically, this will be defined as 1 over nk prime. That's the size of CK prime. Summation for any j in CK prime. I'm going to calculate the distance between I and that point, and then I take the average. That's the second distance. Now I have multiple CK prime, right? Let's say I have capital K clusters. Then besides this cluster CK, I should have K minus 1 clusters. So among those clusters, I am going to take the minimum as the between cluster distance. So I will define the BI as the minimum of all the DICK prime for all the K prime that's not equal to K, because CK is the cluster I is in. So I will call this as the between cluster distance. So this is the between cluster distance. And this AI is the within cluster distance. You can see that the definition of BI is to pick the closest other cluster to data point I. And I'm taking the average distance between I and that closest cluster. So with this, we will focus on AI and BI for our definition of silhouette. So we define the silhouette of data point I, so it's a per data point measure, as BI minus AI divided by the maximum of AI and BI, right? So this definition has a nice property. You can see its sign will carry information. If BI is greater than AI, which it should be for good clustering, then this SI will be positive. Otherwise, it will be negative. So we can actually see that ourselves. So let's just talk about the cases of the order between AI and BI. So let's first think about the case if AI is less than BI. Then the denominator will be BI. Then we will have this as 1 minus AI over BI. So clearly this is positive. And we would also have the other way. If AI is greater than BI, then the denominator is AI, right? And then we can have BI over AI minus 1. And this is what? This is negative because BI is less than AI. Okay, and finally, if AI is equal to BI, then clearly this is 0. So we can actually have the signs to be indicative. And also this denominator can actually make sure that when it's positive, it's less than 1. When it's negative, it's greater than negative 1. So in other words, we can constrain this SI to be between negative 1 and positive 1. So that will make the visualization easier. So then what will be a silhouette plot look like? So the silhouette plot will be something like a bar plot. So it's a horizontal bar plot. So what we will have is that we will have the horizontal axis as 0 here, negative 1 here, positive 1 here. And every row will correspond to 1 data point. So we will have the data point, let's say 1, 2, and here. And it will also tell you the cluster it's in and the neighbor cluster it's in. So for example, data point 1 itself may be in cluster 1, and its closest cluster may be cluster 2. And for data point N, it may be in cluster K, and its neighbor cluster may be cluster 1. So I'm just making up example. So you will have these information and the silhouette. So you would want most data points to have positive silhouette to indicate that clustering is good, right? So you will have a bar for data point 1, and the bar for data point 2, and the bar for data point N. So each bar will go to the number its silhouette is at. So you want to have a lot of long bars to indicate a good clustering. So in other words, the silhouette plot will give you something like a shape. So I'm just making up an example to let you see that. So if the K equals 2, silhouette looks like this. I'll mark it here. So if the pattern looks like, this is for data points. So if this is looks like here, because you have many data points, so you will have quite a smooth shape. That if K equals 3 has here, and also I forgot to say that usually the points will be automatically ordered by their silhouette. So the one with the largest silhouette will be on the top, the one with the smallest will be on the bottom, so the shape will look like nice. And if K equals 3, still negative 1, 0, 1. If the silhouette is it has a larger area here compared to this one, then you would prefer K equals 3 than K equals 2. So this is just a way to visualize, okay, how the overall clustering performance is on all data points. You can see that compared to the WK elbow plot, which only give you 1 number for all data points, this one gave you 1 number per data point. So you can see the overall picture better. So it's possible that you may have some data points, let's say, if I move to K equals 4, it's possible that I may have some data points that have very large silhouette in the beginning, but then it suddenly drops, right? So in that case, you would not prefer a plot like that. So it will give you that overall picture. That's silhouette. Do we have questions regarding the elbow plot and the silhouette plot? You're going to try them on your homework problem. Okay.
So basically, so just a basic clarification: the silhouette plots, the more all the data points are tending towards one, the better it is.
Yeah.
Okay.
Or you can calculate the average silhouette to help you judge.
Okay.
Yeah.
Sorry, I had a question too. So, as K increases, does the average silhouette necessarily increase as well?
No, it doesn't. There's no guarantee for that. Because to answer your question, you need to look at this formula. So it's actually silhouette calculated for one data point. So it's hard for us to talk about the average, but we can look at one data point. So it's possible that when the cluster is not over clustering, so you're not dividing one cluster, which should be one cluster, into two. So in that case, the BI should be much larger than AI. So this data point has a large silhouette. But if you are over clustering, then this data point is actually, so one cluster is arbitrarily separated into two clusters, and then this data point I is in only one half of the new cluster, is in one half of the original cluster. So it's separated from its members. In that case, it's possible that its BI may even be less than AI. So it is on the boundary of the two new clusters. So in that case, the silhouette will suddenly drop toward that point. So that's why you want to, this is a way to justify that this approach is making sense for each data point. That is, it's not necessarily that the more you cluster, the better you have.
Okay, so then you just sort of like maximize the average silhouette, and that's how you pick?
That's one way to pick it. But since we are giving you the plot, you have more information than just the average, right? You can look at the overall pattern. For example, some of you may want to focus on the worst performance. That is, you're not looking at the average, but you want the worst performance to be reasonably good. So these can all be justifications for choosing K. Yeah, so I want everyone to actually bear in mind this general philosophy of doing research. That is, there's not necessarily a unique answer. Your answer may be different from others, but both can be justified. So research papers are also like that, right? You can have multiple ways to do the same thing, but you just need to justify that, okay, it's reasonable. Okay, so that's it. And also, I want to say this. So silhouette can also help you inspect data points by finding out outliers. So it's possible that some data points have very bad silhouette. So in that case, you may want to look into those data points and see what they are, and probably remove them from your data set. Okay, so now I'm going to give you the third approach for choosing K. It's the most statistical one. Just as our name stands, we're going to talk about statistical learning, right? So how do we analyze this or approach this problem from a statistical perspective? This is something I really like, and I want to share it with you so that you can see the philosophy in this approach. And actually, this philosophy can motivate a lot of statistical approaches for choosing tuning parameters in the algorithm. So this is called gap statistic. It was proposed in the 2001 paper by Tibshraeny, Walther, and Hasty. And Tibshraeny and Hasty are professors at Stanford, and they're most famous for their work in lasso, if you have ever heard of it or use it, which we will not cover in this class because we said that we will not talk about supervised learning. So gap statistics is their work on clustering. So the main idea is to think of your data as a random sample from a population. So the first idea we talked about in this class, the the theme of statistical inference. So you always think of some underlying distribution as your population, not just the data points. As you have seen, since we started talking about the clustering, the K-means algorithm itself is not a statistical algorithm because there's no mention of the population at all. You are just operating on your n data points, right? So you're talking about data, you're not inferring anything beyond your data. But the gap statistic has the flavor of statistical thinking by thinking of your data as a random sample. So then the way to choose K is to think, okay, what if my data really comes from a population with no clustering structure at all? Then what K would I pick? So you're trying to construct a negative control for your data by generating a data from a distribution without clustering pattern as a contrast to the clustering on your actual data. So this is the key idea, is to generate some reference data. You can consider this to be the negative control in biology without clustering patterns. And this has a similar flavor to what we talk about as null hypothesis in hypothesis testing. So where the null hypothesis is something as the status quo, but it is something you want to reject. If you reject the null, you make some discoveries, just as the DE gene analysis. So here, when we choose K, we also want to have some null over there. So when we choose K, we know, okay, we are really capturing some meaningful clustering pattern, not just noise. Okay, so here is the idea. So we know that in the elbow plot, we would plot log WK versus K, right? We know that it's going to drop. But if we imagine that we have some reference or negative control data that has no clustering patterns at all, when we perform K-means on reference data and compute the log WK, there must be also a decreasing pattern. Then how can we discern those two, right? So let me say, if this is the pattern you have from reference data, it's also decreasing. But we know because this data has no clustering pattern at all, so the log WK must be larger. So then when you want to choose a K, you want to compare these two trends and see where they differ the most. So here my plot is not very good. Probably I should change it to like this. Okay, so if I change it to something like this, then you can see that if I calculate the distance, let me get a different color. If I calculate the distance for each K between the two curves, then you can see that here the difference is not very big in the beginning, but at this K, the difference, or we call the gap, is the biggest, and then the gap shrinks again. So then this tells us probably we should pick this K as our number of clusters, because this is the K that gives us the biggest difference between our data and the reference data in terms of performance. So that's the basic idea. So then the question becomes, how do we generate reference data? And of course, we don't want this curve to be based on just one reference data. Since we're going to generate reference data, we have the luxury to do repeated sampling to generate multiple sets of reference data. For each set, we can compute one curve, and we want to have the average performance. So therefore, we are going to call this curve as the expected log WK. And this expectation is taken over the distribution of reference data. That is, on average, what the performance will be like on the reference data. So here, when we take expectation, we need to know what is the random variable. Here, the random variable is just WK. It's just the performance measure we computed on one data set. As the data set B is random, then we can take its expectation. Okay, so that's the intuition and the rationale behind the gap statistic. Then the question is how to implement it, right? How can we compute this expectation for each K? So I think the very creative idea lies in the generation of reference data. So the author proposed how to generate reference data. The author proposed two ways, and we can look at them one by one. So the first way is just to take the original data. So of course, you want the reference data to resemble your original data as much as possible, but not having any clustering pattern. So that's the goal for generation. If the reference data do not resemble real data at all, then this reference is not a meaningful reference. Just like in your biology experiments, when you design a negative control, you just want to make the variable you care about different, and you want to control the other variables to be the same. So you're not having confounder effects in your analysis. This is the same idea. So the first way is to define a domain, which is like a rectangle, or it's a rectangle in P dimensions. Define a domain of n data points, and then do uniform sampling to generate reference data. So let me give you an example for P equals 2. So let's say that you have n data points with only 2 dimensions. So if the data points looks like this, so clearly you have 2 clusters. And to generate reference data, what you want is to define the limits for the minimum and the maximum in each dimension. So let's say that you have this as the minimum in the first dimension. This is dimension 1, and this is dimension 2. So let's call that AB and CD. So then this gives you a box, right? A rectangle defined by the limits. Then what you're going to do is that you're going to draw n data points from this rectangle uniformly. So then the reference will look like so you're going to keep this rectangle and you're going to draw n data points just uniformly. Yeah, so n data points. This is the first approach. And of course, uniform sampling will not give you any clustering pattern. So then you apply K-means algorithm to this data, and you can get a log WK. And you repeat this sampling, you get another log WK. And so let's say that you do that for capital B times, you will have log WK1 for the first sampling, 2 log WKB for the B subsampling. And then you will compute their average. And you will call this as your expectation, if your B is large enough. So you're basically approximating this. And later, I will tell you that this has a name, this approach. Using repeated sampling, taking the average as the expectation, this is called Monte Carlo method. So you're using repeated sampling to approximate expectation. Okay, so that's V1. But one issue we will have here is that it's possible that the generated reference data do not resemble real data at all, right? So we see here the real data are only lying in the two corners in the rectangle, but here it's everywhere. So they're not really similar. So to make it more similar, the author proposed a way 2, which I think is very good to know. And this is related to what we call PCA. So way 2 is basically to recombine the original P dimensions into more meaningful dimensions. And then you are just going to generate data in that new coordinate system corresponding to the new dimensions instead of the original dimension. So in the new dimension, we want to have the first dimension capture most of the variance in our data. So clearly in this case, it will be this diagonal direction. That will be our new dimension 1. And what's orthogonal to it is the new dimension 2. So this can help us define a better rectangle for generating data instead of the original one. So that's the intuition behind that. So, and that's just the idea of PCA. So to really understand that, we need to have some basic knowledge about what we call the singular value decomposition. So singular value decomposition is a very useful way for extracting the meaningful information in a matrix. So here the matrix, if we put it as a, so let me just try to say it. So if we put it as an n by p matrix, okay? So you have n data points. Each data point is p dimensional. So the columns will be p dimensions, and this will be data point 1 to n, corresponding to n rows. So if we stack our data in this way, so what does the singular value decomposition do? It will find, and also please note that works for any matrix of any dimensions. So it will find a U matrix, which is n by k, times a diagonal matrix, which is k by k, and another matrix which is often called V transpose. And this V transpose itself is k by p. So in other words, here this diagonal matrix contains what's called singular value, and the singular value can be ordered by their magnitude. So the largest singular value will denote the most important new dimension to the kth singular value for the least important new dimension. And so what you are going to use is this U matrix, okay? Because this U matrix is basically what is the n data points in those new k dimensions. So you're going to encode your points in k dimensions as opposed to the original p dimensions. And this other matrix is a k by p matrix. It's about how each new dimension, each row, corresponds to the original p dimensions. So here we would have each row as length p. So that would be the weights of the original p dimensions, maybe p genes, for example, in this new matrix, in this new dimension 1. So this can actually provide us with a very useful way for transforming our data. And that's just the idea of PCA. So in fact, if we know what is PCA, maybe I'll give this preview here, but we'll talk about this in more detail later. So in the PCA, it's only applied to a squared matrix, okay? So it has to be a squared symmetric matrix to have positive eigenvalues, non-negative eigenvalues. So the PCA is applied to what we call X transpose X. So you're going to multiply X transpose to the left of X this will be a P by P matrix, okay? And if you look at the singular value decomposition, take it as it is, then this will become, we take the transpose of this one, so we will have V times, and D transpose is D because it's diagonal, and U transpose, okay, that's X transpose, and then X, U, D, V transpose. So one property about singular value decomposition is that in the U matrix, all the K columns are orthogonal to each other. So that means if you take U transpose U, you will get an identity matrix with K dimensions. And similarly here, if you do V transpose V, I will get an identity matrix with K dimensions. So then you can see, after combining these two terms, the PCA will give me V times D squared, which means I square every element in the diagonal element of D times V transpose. This is what we have. This is what we call as Eigen Decomposition. So in linear algebra, Eigen Decomposition is for a symmetric matrix. It doesn't work for those matrices for square matrices. It doesn't work for non-square matrices. But singular value decomposition is more general. It can be applied to any matrices. So then you see here the V is still this V. So in the Eigen Decomposition, what we keep are the column singular vectors. And now let me mark the dimension. So this is P by K, this is K by P, and again, this is K by K. So each row here, or each column here, corresponds to one of the K dimensions. It's a linear combination of the original P dimensions. So this in PCA is what we call the loading matrix, which we'll revisit later. This is what we call the loading matrix, and this one contains what we call the eigenvalues. So by the relation, this is D squared, this is D. So you can see each eigenvalue is the square of a singular value, but both still carry the magnitude. The larger the eigenvalue, the more important the corresponding new basis is. Okay, so this is providing what we call the loading matrix. It's about how each new basis corresponds to the old P basis. But this U here is what we will need for the embedding. And so we can say that if we can call this new, the two, let's say still in P equals 2, okay? So let's revisit this example by setting K equals P equals 2. So we're not doing any dimension reduction. We just do some rotation. In that case, this U will correspond to two columns, and we can call them as PC1 and PC2. So therefore, it's basically a rotation. So as I said, in the new dimension, we know the first meaningful dimension will be this way, and the second one is orthogonal to it, so it's this way. So therefore, the data points would look like this. It's after rotation. So then I'm going to define the limits in this new dimensions. So I will have a limit here and a limit here. So that would be my new matrix, sorry, new rectangle. So I'm going to stay in this PC1, PC2 dimension, and I'm going to draw uniform sampling from this matrix. Okay, so I'm going to do that. After uniform sampling, then I need to rotate the matrix, the dimension, the space back to the original dimension. So essentially, from the original dimension to this one, I skipped that, but I can say that it's basically by by right multiplying this v. So basically, I can do x times v, okay? And that will actually give me, this is equal to u times d. So it's actually these two parts. I can do the right multiplication of v. So v transpose and v got canceled. So this is what I'm plotting here. And with this, I can do this uniform sample in the PC space. Then I want to rotate it back to the original dimension 1 and dimension 2. That's what I want. So then how do I do it, right? So right multiplying v can convert the original matrix, the original space to the PC space. How about if we want to go back from the k dimension new space to the p dimensional original space? So again, you just need to multiply v transpose, right? Multiply v transpose again. So then what you do is that taking this matrix, probably let's just call it some, I'll just call that Z matrix, okay? This Z matrix will include my new data points. So it will be n by k. So here k equals 2, but it's basically my new data points in the rows like here. So then what I would do is that I do Z times V transpose. So this is to rotate this k-dimensional space back to the original p-dimensional space, because this part is k by p, and this is n by k. So you can see I'm getting a n by p. And then this rotation will restore the original direction. So then I would have the data points to be like this after rotation. Okay, then I'm going to take this as my new reference data. So this will be the thing I'm going to apply my K-means algorithm to generate this expectation. So you see that PCA or SVD is a very magical thing. It can help you get the more meaningful dimensions from your original matrix. And as we'll see later, this is especially useful for gene expression data, because we know over there the P dimensions are P genes. And we know that many genes are highly correlated with each other. So therefore, they may just be combined and represented by one new dimension. So that's a very useful way for data analysis. And we'll talk about that later. But this is just a very interesting use for a PCA for generating reference data. So that you can see that here the reference data, although it's still having no clustering pattern, but it resembles the original data much better than the reference data generated in this way. Okay, do we have questions about this part so far? So even though this might be something new to you, like the SVD PCA, but I hope you can understand the intuition about what it is actually doing. So I would say for linear algebra, I know it's very difficult it's taught as very difficult in many classes, but I would say the key to really understand what linear algebra is doing or what the matrix is really doing is to think of the matrix as a linear transformation. So, or linear map. So let's say, again, in this matrix, let's say it's a K by P matrix. So you can consider the columns to correspond to a space with P dimensions and the rows to correspond to a space with K dimensions. And if you have a matrix that is, or I should say, yeah, I'll just say that this Z matrix, okay, so it's K and it's N here, just our example. And this is what we call V transpose. So in our example, you can consider this Z just to encode N data points in a K-dimensional space. And what this matrix is doing is that by doing the right multiplication, you are going to map the same N data points to P dimensions, to a new P dimensional space. And then this transformation or this linear map is just telling you what each new basis in the new space corresponds to in the original k-dimensional space. So each value here of the k value here is a weight. So it's just how much contribution each of the k bases make to a new basis in the p dimension. So we'll have p columns for p weight vectors. So it's basically a transformation. So after the transformation, you still have n points, but each point now has p coordinates in the new space. Previously has k coordinates in the old space. And we're using that property for mapping the space from the original space to the PC space and back to the original space. And this is very useful in bioinformatics because nowadays we analyze most data in matrix forms. So understanding this nature is very important. Okay, do we have questions about this part? Okay, so now let's talk about the gap statistic algorithm. So the algorithm has an R package implementation. So you're going to play with it in your homework. So of course, to choose K, we need to set a maximum for our possible case, for our candidate case. So we will need a K max. We will not go beyond that because for each number less than this K max, we're going to compute its gap statistic. So, by the way, the gap statistic for K, I should say it here. So we always carry the sample size, number of data points here with us because this sample size will also be used for reference data generation. So we will evaluate this gap statistic at a particular value K. And it's defined as 1 over B over the generation of reference data, B from 1 to capital B, log WKB. Okay, this is the average of reference data performance minus log WK on our real data. So in other words, it's reflecting this gap we have seen here. So the reference expectation minus the actual performance. And we want to find the smallest K to maximize the gap. That's our hope. So then in the algorithm, what we do is that for each K, we can of course start from 1. Right? Because if you ended up choosing K equals 1, you will not do clustering at all. It means the data doesn't have any pattern for you. 1, 2, up to K max. And then what we do is that we apply K means to a reference data, right? To reference data set B, and that will give me log WKB. And I will repeat this for capital B times. And then I just calculate this and I'm done. So this will give me, so after all of this, and I will also actually say apply k-means to original data, and that will give me log WK. Okay, then I will return this gap NK for this particular K. So we'll have that for gap N1, gap N2 to gap NK max. So now it's the choice. Which K do we choose? So one nice thing about this gap statistic is that they also considered the uncertainty in the estimation or in the estimation of this, let me put it here, in the estimation of this expectation by the Monte Carlo method, by this repeated sampling. Let's say that your capital B is not very big because of computational issue, right? You cannot afford to run the k-means for so many times if your n is very large. So in that case, you need to consider the uncertainty over there. So that's why there's also a standard deviation associated with each k. So I'm just going to skip the detail there, but this standard deviation is about the uncertainty in repeated sampling. Uncertainty in reference data sampling. So in short, we should say that as the B goes up, the standard error of K will go down. So that's the relationship. If the B is very small, then you have a larger standard error in your gap statistic. So then for choosing K, the authors apply something called a one standard error rule. This is a very common rule of thumb. What it means that when we choose the largest gap, we're not really targeting the largest number here. We also consider the standard error. So for example, if the previous, because we always prefer a smaller k, right? For that's just what we call the Occam's razor. It's just the principle of choosing the simplest model, or the another way of saying it is just the sparsity principle. So you want to prefer a simpler model than a complex model. So in the context of clustering, more clusters is more complex than fewer clusters. So because of that, to choose a smaller k, if a smaller k has the gap statistic value which is smaller than its next k plus 1's gap statistic value, but not too small, if their difference is within one standard error, one standard deviation, we pick the smaller k. So that's just a rule of thumb for choosing the simpler k or smaller k. So that is to pick the smallest k such that gap at k is greater or equal than gap n at k plus 1 minus SD k plus 1, right? So for each gap statistic, we carry its error bar. So basically what this means is this. Let me give you a graph. Okay, so if we have gap k here, gap k plus 1 here, but the error bar is plus minus 1 SD. So since this gap k is larger than the lower bar of gap k minus 1, k plus 1, so we pick this one. That's just what it means. You're comparing each number with the next k's lower bar, not just the next k. So that's by considering the uncertainty in the calculation of the gap statistic. And again, this uncertainty lies in reference data generation. Okay, I just want to conclude this part by saying that the use of uncertainty in choosing a tuning parameter is a very useful idea to know, because in bioinformatics analysis, most of the time our conclusions, our discoveries are made based on our particular data set, data we collected from experiments. It's not really, we don't know what the true distribution or population is about, right? So, but still in that case, it's always useful to introduce some perturbation to your data by shuffling some data values, by adding noise, or by downsampling your data as a way to check the stability of your procedure, to make sure that what you find as discoveries are not a special case. So it's actually a pretty robust finding from your data. So that has the same essence as the gap statistic idea here, by having some random control from your data. Okay, do we have Other questions before we conclude this part? So that's the last thing I want to say about choosing K. So in the notes previously, I mentioned the jump method, but I think I'm going to skip it for this year, since I think that one is less widely used, and also it has stronger assumption by assuming the data follows some multivariate normal distribution. So this one doesn't have any assumption on the data distribution. Okay, so that's it. Then the next method, clustering method, what we'll talk about is what we call the hierarchical clustering. So it's a method that's very widely used in heat map. In the heat map function, by default, it will give you the hierarchical clustering of the rows and columns. So we're going to talk about how it actually works. I would say hierarchical clustering is older than K-means clustering because it's less computation intensive. You can actually do it by hand, and you're going to do it in your homework. So what's different between K-means and hierarchical clustering is that, by the name, hierarchical clustering means that you have a hierarchy of clusters. So at the root, you have only one cluster at all. And at the bottoms, at the root, at the leaves, you're going to have every data point as its own cluster. And you're going to aggregate data points gradually until they are merged into one cluster. So this is also called as agglomerative clustering. Same for hierarchical clustering. So for hierarchical clustering, what you need is two distance metrics. So you will need a distance metric between data points. So for example, Euclidean distance or the L1 distance, just as the summation of the absolute differences in dimensions, in every dimension. We also need a distance metric between a point and the cluster. During our aggregation process. Right, so after we combine, let's say, 2 points into one cluster already, then we need to further decide whether we're going to combine this cluster with another point or we keep it as it is. So that decision will depend on how we measure the distance between a point and the cluster. So there are 3 ways of defining defining this distance. The first one is what we call single linkage. So in single linkage, let's say we define, okay, so this can be generalized to between a point and the cluster and between 2 clusters, right? So this is even more general because a point itself can be regarded as a cluster. So we are going to talk about the more general version, how to define a distance between 2 clusters. So let's just say we have clusters A and C. Okay, 2 clusters. Then the single linkage is defined as, I use the subscript SL for single linkage. Between A and C, it's defined as the minimum distance between points. Okay, I'm going to use that previous notation just for your convenience. Xi and Xj, where I is in A, J is in C. So I'm going to take pairwise points, one from each cluster, calculate their distance. And from those pairwise distances, I take the minimum. I call that as the distance between the 2 clusters. So intuitively, so maybe I can say that I can use a plot to help understand. Still, I'm going to talk about the case when P equals 2. Because that's when I can draw data points. So if this is cluster A, okay, and if this is cluster C, so single linkage is going to pick the closest distance. So here by eyes, you can see this one will be what we call as the single linkage, the pick it the closest one. And in contrast, we can pick the largest one, right? That is called complete linkage. So for complete linkage, we define it as the max. I in A, J in C. So in our plot, I'm likely to pick just, yeah, I think probably I'll just pick this one. Okay, the farthest two. I call that complete linkage. And lastly, average linkage, or this is also called group average. So this is to take the average of all the points in A, all the points in C. And I'm just going to do the double sum, size of A, size of C, right? I in A, J in C. And I take the average of pairwise distances. So we have three different ways of defining the distance between two clusters. And as you can imagine, they will give you different results. So that's why when you use hierarchical clustering for your research paper, please make sure in the method section you specify, okay, when I do hierarchical clustering, I use single linkage, I use complete linkage, or I use group average. I think in R, the default is complete linkage, if I remember correctly, if you don't change it. So this will determine how we do aggregation. So the idea of hierarchical clustering is very simple. Basically, we start from all data points, n data points. From these n points, we pick two that's closest to each other. We merge them into one cluster. So now we are having n minus 1 data points, right? Because one point represents a cluster. Or I should say n minus 1 data, n minus 1 clusters. Then among those n minus 1 clusters, I find the closest two again, merge them, and then I get n minus 2 clusters, and so on, until I have only one cluster. So every step, I only reduce the cluster number by 1. So once we return to the class next time, we're going to give you a concrete example with 5 data points to actually do this ourselves, so we can see how single linkage works. And also single linkage, for your information, has a close connection with a popular algorithm called minimum spanning tree. So it's an algorithm for connecting data points into a tree structure, just like what we did in hierarchical clustering. So there is a research paper that discusses the connection between those two things. So I'm going to post that to Cecily for your information. So minimum spanning tree has been a very widely used algorithm for pseudo-time inference for single-cell RNA sequencing data. In other words, you have n cells and you want to connect them into a trajectory. So that based on that connected trajectory, you want to say, okay, these cells corresponds to earlier developmental stages, these corresponds to late stages, and I can see how the cells change along the trajectory. So I can analyze what genes has expression changes in what way along the trajectory. So I can talk about some new functions of those genes if I don't know of, right? That's a very popular analysis in single-cell RNA-seq data, which is your project data. So that's all for today. And at the end, I want to also say something about the project. So some of you asked me what kind of project we are looking for, or what's the expectation of the project. So I want to say what I care the most would be your thoughts, your innovation. So how much thought you put into the data or relevant literature to really formulate an interesting question. So in past years, we have seen questions that are very biological. For example, a student studies neurobiology, and so he or she tries to find some genes relevant to neurobiology and analyze how those genes are expressed in those cells. That's one type of problem. And another type of problem is to look at the technology. So we know that data set contains, this is a benchmark data set that contains multiple experimental protocols. So it will be interesting to see how much do the protocols affect the data quality or the biology you can learn from the data. That's another type of problem you want to try. A third type of problem that can be asked is about computational methods, right? So for, in the single cell field, if you search for a single cell computational method, there's a website that hosted, I would think, around 100 methods, or even, actually around 800 methods for many tasks. So for one particular task, there are many methods that has been developed. So if you are really interested in how these methods for the same task work on one data set, that can also be a problem to work on for this final project. So in other words, there are many things you can explore with this data set. And my suggestion is to start early, to find your partner, or if you decide to work on yourself, just start early. So then you can, but we can actually see your efforts in the final presentation and the final reports. If you want to discuss your ideas, you can come to my office hours. Okay, so that's all I want to say for today. And for those of you who want to stay for discussion, please feel free to do so, or please feel free to leave.